and thank you to the organizers for giving me an opportunity to present this work today. So the blood-brain barrier, or the BBB for short, is important for normal brain function. It supports neuronal function and protects the brain from harmful substances. However, this protective role also makes it an obstacle for drug delivery to the brain. The regulation of the BBB is complex. Endothelial cells, or ECs, here shown in purple, comprise the brain blood vessels and are the site of the BBB in mammals. Brain ECs have specialized properties that promote low blood vessel permeability. However, BBB properties are also dependent on factors from the cellular microenvironment. So transplantation experiments have shown that the brain environment contains factors that induce BBB properties. And to determine the source of these cues, several studies have looked at the role of perivascular cells, such as pericytes, here shown in teal, and astrocyte end feet, here shown in blue, finding that they are important for the formation and maintenance, respectively, of the blood-brain barrier. Interestingly, it's also been observed that different brain regions show different BBB properties. On the extreme end of this heterogeneity are the circumventricular organs, which are small brain regions that have leaky BBB, despite being in the context of brain pericytes and astrocytes. The leakiness of these regions is essential for their function, which involves communication between the brain and the periphery, and they regulate processes such as hunger, thirst, and blood pressure. Here are images of a tracer leakage assay, which we use to assess BBB permeability. This mouse here was injected with a tracer and then perfused to remove excess tracer from the bloodstream. So this is a section of the cortex, and as you can see, despite the abundance of blood vessels, the tracer was retained within the vasculature by the blood-brain barrier. In contrast, in ME, one of the circumventricular organs here found at the bottom of the mouse brain, it's a small brain region, you can see that the tracer leaks out of the blood vessels and into the brain area. This image was taken using a 3D tissue clearing and imaging method, which was developed in the lab of Zhuhao Wu at Mount Sinai School of Medicine. He's our collaborator. It's an aqueous-based method. And it was performed by Urs Langen, who's a postdoc in my lab who I've closely collaborated with on this work. And all the rest of the images, the fluorescent images in this talk, were produced by him using this method. Okay, so we hypothesized that differences in brain endothelial cell function, together with local differences in the BBB environment, are what modulate regional barrier function. And we sought to leverage the natural differences in the BBB, so between the cortex and, in our case, the median eminence, to try to find differences that would naturally modulate barrier properties. And so together, using single-cell RNA sequencing with this imaging method, we were able to both look at rare cell types in this small brain region and also map them back into, in the case of the ME, the entire brain region, and then in the cortex, the size match region, to really understand the molecular correlates between the data. All right, so we performed in-drop single-cell sequencing using cells from both these areas in adult wild-type mice. As you can see from this T-SNE plot, we identified all major cell types in the brain, and importantly, endothelial cells, pericytes, here made of mural cells, and astrocytes. When directly comparing cells between these two regions, we observe some clusters that contain cells from both regions, as well as some that are unique to each region. Interestingly, astrocyte populations between the two regions are quite different, and we observe an ME-specific population of endothelial cells. We next look more closely at endothelial cells. They are the site of the BBB, and the ME-specific population that we see expresses a known marker gene. We expected to see this based on previous histological analyses. We compared this ME-specific EC population to cortex ECs, and we found approximately 400 differentially expressed genes. And these included genes that were important for the BBB, such as tight junction components, transporters, immune molecules, and transcription factors. One such gene, endomucin, had increased expression in the ME-specific endothelial cell population, and using uClear, we were able to validate the expression of endomucin, increased expression of endomucin in the ME compared to the cortex. We did this for a number of other genes as well. Additionally, using nuclear, we were able to observe striking differences in endothelial cell morphology. So just looking here at white, this is a blood vessel marker, endothelial marker. You can see the morphology looks quite different. We quantified these data and found that there's increased endothelial cell density as well as increased capillary diameter. Beyond these clear differences in endothelial cells, we also observed some differences in parasites and astrocytes. So here are the parasites. We used a parasite fluorescent reporter to create 3D models of the cell morphology. And you can tell comparing the ME with the cortex, these cell morphologies appear to be quite different. However, our sequencing data did not show an emergence of an ME-specific parasite population. So the parasites are here in pink. And when we look at it by region, there's no obvious emergence of a specific population. We do see a fibroblast population that appears to be specific to the ME. I'll get back to that a little bit later. So currently we're still investigating this. We don't know if this is the true biology of the situation or if this is simply undersampling. We do have noticed that there are fewer parasites derived from the ME compared to the cortex, and we can validate this in our imaging data. So we see a decreased parasite to endothelial cell ratio in the ME compared to the cortex. Next, morphological analyses with astrocytes using nuclear and an astrocyte-specific reporter revealed clear differences in astrocyte morphology between these two regions. So the cortex astrocytes, here in red, have the classic stellate morphology. They interact with multiple blood vessels. But in the ME, we noticed two different populations. So one was this vessel-associated population, seemed to be nestled in with the blood vessels, as well as a ventricle-associated population, so this cell here, which seemed to be more associated with the nearby third brain ventricle. This is not terribly surprising given the known heterogeneity of astrocytes throughout the brain, but we sought to find molecular correlates between these different populations in our dataset. So we found two main populations of cortical astrocytes and ME astrocytes, as well as an additional GFAP high population. Some genes that are enriched in these populations are shown in this heat map. We confirmed expression of several of these genes, including ALDH101 using fluorescent reporter lines. So from the heat map, you can see that ALDH101 should be expressed in both cortical populations, and that's indeed what we see, as you can see by the GFP signal here. However, in the ME, only one, the ME1 population, should express it, and we do see that this seems to label a subset of the astrocytes in the ME region. These differences between endothelial cells and astrocytes, together with the fact that enriching our differentially expressed gene, we find that most of them are many of them, approximately 70%, are associated with functions of secretion or localization to the cell membrane, suggests that perivascular cell communication is likely different between these two regions. To kind of bridge the gap and I guess build more evidence for this, we focused on looking at these intercellular interactions using transmission electron microscopy. So just to orient you to these images, the astrocytes are shown here in light blue, parasites in teal, and the blood vessels, the endothelial cells, in purple. So what you'll be looking at is a cross-section of this. And then in the cortex, this is a well-known interaction. The endothelial cell here is interacting very closely with the parasites, and astrocyte end feet are very close nearby, interacting with the cells. In the ME, the situation looks a little bit different. So the endothelial cell here still interacts closely with the parasite. However, it seems to be embedded in a thick extracellular matrix. Astrocyte end feet, we know from our fluorescence data, are present in some way, but they're not very easily distinguishable in the brain parenchyma. And we also noticed the presence of fibroblasts in BCN, so here highlighted in red. So to investigate the intercellular relationships between these, we performed region-specific expression analyses to look for intercellular signaling. We used a method developed previously by Kumar and colleagues. We assembled a ligand receptor database, which included well-characterized pairs listed by the original authors, as well as some that we found to be differentially expressed in the ME and cortex ECs, parasites, and astrocytes. We calculated an interaction score by looking at the expression of these two molecules in candidate signaling cells and receiving cells. So here I'll show you endothelial cells and parasites and endothelial cells and astrocytes. And we're looking for pairs with molecular complementarity. Using this method, we find some pathways that we expect. So between endothelial cells and astrocytes, here we see enriched VEGF signaling. And in endothelial cells and parasites, we see an expression of the PDGFR beta, PDGFB signaling pathway. Additionally, when you compare them side by side, we do see differences between the two regions. This is an area of ongoing investigation, and we're hoping that these molecular analyses will help serve as a stepping off point to further dissect how each cell type specifically contributes to BBB heterogeneity. So with that, we observed striking molecular and morphological differences in endothelial cells, parasites, and astrocytes between the ME and the cortex. And we detected regional differences both in intercellular interactions as well as the expression of intercellular signaling molecules. And in conclusion, looking forward, we hope that understanding how regional BBB heterogeneity is achieved may help us better understand regional brain functions and may allow for the development of more effective, potentially region-specific targeted therapies. With that, I'd like to acknowledge my advisor, Qinghua Gu, Urs Langen, who I mentioned previously, the postdoc I collaborated closely with, other members of the GU Lab, our collaborators, the core facilities at HMS, and my funding source. So thank you.
Thank you very much. That was really interesting talk. Maybe I'll start with my own question. We know in cancer, the vessels tend to be leaky. Are there any changes relating to the pericytes and endothelial cells and their interactions that somehow parallel changes that are seen in tumors?
So the ME vessels are fenestrated, which is a huge difference between the BBB containing cortical vessels and the ME. So I'm not sure exactly how the changes are in cancer with regard to fenestrations, but yeah, so the parasites...
Basically separation between the parasites and then they become separated. So the attraction becomes lower.
Interesting. Yeah. Yeah.
Thanks.
Very cool. I'll ask another one. I was wondering, as you're thinking about these differences in interaction and the structure of sort of the astrocytes, how dynamic these are, and like what kind of changes do you see in some injuries? I know Shalep was bringing up the idea of cancer. I was thinking about, strokes and various things of that nature. I was wondering more about, how static this was versus how dynamic. Yeah,
so we're not entirely sure how dynamic the heterogeneity is. However, in the ME is characterized as a quite dynamic region. So it does regulate processes such as hunger. And so there are known changes in signaling between the ME and hypothalamus respond to hunger cues. So it is likely that, at least in the ME, this population is potentially dynamic. Some people propose that there are, it's been seen that there are endothelial cell loops that kind of respond to these things. So yeah, we've only looked at one homeostatic basal condition, but it would definitely be interesting to test in a more dynamic condition. And of course, in disease would be very interesting as well.
Awesome.
Great. We have another question from Zhu. A very cool talk for the fibers associated with the ME. Do you know when they arrive during development and what is their origin?
I actually don't know that. I'm not sure. But I do know that they resemble, looking at the EM, actually, it kind of resembles the choroid plexus in some way, which is another permeable region in the brain. But yeah, beyond that, I'm not entirely sure of the developmental origins, but it would definitely be interesting whether it was sort of more central or peripheral.
And can I ask to elaborate a little bit on the role of the astrocytes in the whole story? So I understand the endothelial, the pericytes, but how do the astrocytes come into the equation?
We're not sure. So it's very weird that in the ME, they're not interacting as closely as in the cortex. I think in the cortex, it's thought that the proximity of them is essential to the signaling and the regulation of potentially processing of nutrients and stuff like that. In the ME, the fact that they're more removed or one that's really closely associated into the vessels kind of suggests that they may function differently. We also wonder what the role of the prominent extracellular matrix in the area is. I think there are a lot of questions that have opened up looking at this morphological and molecular analyses that we're excited to pursue.
Thanks.
Very cool. And Sanjay asks, one more question. He says, great talk. What is the molecular weight of the tracer? He says, I might have missed this, but do the astrocytes singly associated with these regions have different developmental histories?
So the tracer that we use that I showed here is NHS sulfobiotin. So it's a small molecular weight tracer. I also am not sure about the developmental origins. I should look up this of the astrocytes. But yeah, it would be interesting to see if they stratify in some way.
Okay, thank you very much.